Good afternoon, everyone, and welcome to today's Applied Plant Sciences seminar. Today, our speaker is Michael Burns, who will be talking to us about his project on identifying and quantifying kernel attributes affecting quality in Masa-based products. So a little bit about Michael before he starts. He grew up on a corn and soybean farm in Watertown, Minnesota, which is about 45 minutes west of the cities, and he attended University of Wisconsin River Falls for his bachelor's degree in biotechnology with a minor in chemistry. And while he was there, he was able to participate in some cool projects, including genetic screens for honeybee mating, developing a plant transformation lab for a plant tissue culture class, and data analysis on earth-kind rose trials. And he also spent 2 summers working here at the U in Jerry Cohen's lab, looking at how media affects auxin production, as well as learning how to repair various analytical tools. And through all of these experiences, he became motivated to come to graduate school so that he could learn new things and get to explore interesting and the sometimes strange research questions that we get to learn about. And when he's not working, he enjoys spending time outdoors with his wife, running and working on Raspberry Pi projects. And without further ado, I will turn things over to Michael for his seminar.
Thank you so much, Julia. As Julia talked about, I'm going to talk to you guys today about identifying and quantifying kernel attributes affecting quality in Masa-based products. And a quick disclaimer, this work was funded by PepsiCo Incorporated, and any opinions or scientific interpretations expressed in this presentation are those of the author and do not necessarily reflect the position or policy of PepsiCo Incorporated. A brief outline of what we're going to talk about today. We'll start with a little bit of background on food grade corn. We'll talk about the significance of the projects that I'm going to propose. Then we'll get into my projects of predicting moisture uptake and pericarp retention. We'll talk about some biological questions related to these 2 projects. And then we'll end with a question and answer section. So first, a little background on corn. Corn is America's largest crop. As of 2019, roughly 87 million acres were grown, and in 2020, 92 million acres were grown. As you can see, Minnesota is the 4th largest production state of corn, and it really affects the economy here in Minnesota. Now, corn is used for a wide variety of purposes. The 2 largest industrial segments being feed and residual, as well as fuel ethanol. But we're not going to be focusing much on that. We're actually going to be focusing on this blue little sliver down here, which makes up about 1.5% of the corn grown in the US. This is the food grade corn. And while it may be only 1.5%, 1.5% of 92 million acres is still roughly 1.4 million acres. It's a very substantial land use, and the industries that use this corn are actually quite large. The tortilla chip industry alone is worth approximately $20.2 billion globally. So there are a lot of different varieties that food grade corn can come in, some of which you may recognize. I want to go through 4 examples with you so that we can better understand what I'm going to be focusing on. First is everyone's favorite movie snack, popcorn. Popcorn is special because of its thick pericarp, which allows it to build up pressure while it's cooking. Going to the other side is sweet corn, which actually is a thin pericarp, and you actually eat it at an immature stage, so it's still very fluid, very high in sugar content. We also have flint corn, which some of you may know for its colors. If you've ever had grits, this is a very common corn to use. It's very dense and hard. And we also have dent corn. This is a very common corn to grow here in the US. It's most likely what you see when you drive by a field of corn. And this is what people use to make things like tortilla chips or corn chips here in the US. And so we're going to be focusing on more of the hard corns, not so much the popcorn or the sweet corns. We're going to be focusing on products actually made of dent corn, though flint is used in some of these products. But some of the products that they make are things like cornbread, tortilla chips, cheese puffs, tortillas, hominy, and tamales. And we're actually going to focus even more exclusively on these three here. They're made from dent corn, and actually more specifically, they're made from Masa, which we will get into very shortly. But before I can actually get into what Masa is, I need to do a little bit of a review of kernel composition and anatomy. Now, a corn kernel is made up of roughly 62% starch, 19.2% protein and fiber, 3.8% corn oil, and about 15% moisture by weight when it's dried out and ready to be processed. Now, these constituents, the oil, the starch, and the protein and fiber, are all placed in different locations of the kernel. You have the pericarp, which is sort of a thin layer of tissue surrounding the kernel. You have the endosperm, which is the energy source of the kernel in the center of it. I mean, as you can see, these arrows are pointing to two different regions. You have a hard endosperm and a soft endosperm. And finally, you also have the germ. And it's important to recognize these three locations because they all have a different compositional makeup, which will affect how they are processed during, or how they're affected during the cooking process. So as you can see here, pericarp is made up of primarily fiber and protein. Endosperm is made up of primarily starch and protein, and the germ is the most oil-heavy location of the kernel. So getting back from the composition, now that we actually understand the composition, we can understand how it's affected and how it affects the quality of these cooked products. So getting back into what Masa is made from nixtamalized corn. And so here on the left, we show a picture of raw kernels. On the right, we show pictures of corn being cooked. And in the middle is a spoon, which shows an example of both next to each other, so we can see a little bit more comparison in more detail. And so as you can see, the raw kernels here really aren't affected. They look as they do in the picture in the upper left. But if you look at the kernels to the right, they kind of look fuzzy, almost slimy. And this is because the pericarp is actually starting to be removed. The nixtamalization process is great for removing the pericarp and adding water into the endosperm to sort of soften it up and facilitate grinding. And so this nixtamalization process is actually a quite long process. I want to walk you through this before we get going too fast on any other steps. So in an industrial setting, nixtamalization, let me back up. Nixtamalization is a thermal-alkaline cooking process, meaning we're going to heat it up and we're going to raise the pH. So at an industrial scale, what this looks like is corn will come in a large batch. The manufacturer is going to add water, probably start heating it at around this stage. They're going to add lime, calcium hydroxide, to increase the pH of the solution. They're going to heat this and let it steep. And by steep, I mean similar to when you make tea. It's where you let the kernels sit in this warm solution for an extended period of time. In our case, the cooking process, the heating, takes anywhere from 5 minutes to 180 minutes, depending on the product. The steeping phase takes roughly 16 hours. Following this steeping, they're going to send it through a process to remove the pericarp. Generally, this is done with water jets. And we want most of the pericarp to be removed, but not all of it. And that will come up more later as we talk about my second project. Following the pericarp removal, the samples are ground between two stone mills. And this creates a flour that's very light and fluffy. It has a lot of air pockets, which isn't necessarily great for cooking. It's not going to cook very uniformly. And so we put it through a process called extrusion, where we send all of this mass through an extruder, which has a fixed cross-sectional area, and then condenses it all into this dough that we call masa. So masa is the end product, the end dough product of nixtamalization. And this is what we're going to be focusing on. So a lot happens during this nixtamalization process. As I kind of alluded to earlier, the pericarp has a big effect or that undergoes a major change, and that change is removal. As you can see, the pericarp has been completely removed in this scanning electron micrograph, and it's not the only thing to change. In the endosperm, you have these starch granules that, as you start heating them up in a water solution, will actually swell and disform and sometimes burst. And both of these chemical reactions actually have a pretty big effect on the cooking quality of the masa. And so, for example, I just wanted to take a moment to explain what kind of zone we want these traits to be in. We don't want the moisture content, for example, to be too high or too low. Too low of a moisture content will lead to poor cohesiveness and poor texture. Too high will lead to a high oil content in the chip, and a high moisture content could also lead to a really sticky masa, which isn't going to be very cookable. What we're actually looking for is something in the middle that is in that just right green zone. And this will change depending on the product. Typically, when we're talking about corn chips, it's going to be right around 45%. But again, that will change depending on manufacturer, specific product, things like that. And there are many qualities to making a good chip that people need to be concerned about. So when you make a good corn for chipping, we're really worried about things like yield, test weight, stress cracking, disease prevalence, and kernel shape. And these are all going to be things that the breeding company will test for. And they do these on most lines anyways. They really focus on the stress cracking and test weight for food grade varieties. However, these bottom three that you see, the moisture uptake, pericarp retention, and these other various tests that are important to consumers are going to be actually tested at a pilot plant. And so we're going to focus primarily on the moisture uptake and pericarp retention, as these have actually effects on flavor and mouthfeel in other products. But the problem with doing this setup is the pilot plant is just too low throughput for breeding companies. You know, they can't get a lot of samples processed fast enough for these breeding companies who have hundreds or thousands of lines to test. And so they really need a faster, more high throughput method to analyze these. So breeding companies have kind of come up with some proxy tests to test for cooking quality. The kernel density is largely correlated to cooking quality. And so they'll test for kernel density through like tests where they test how many kernels sink or float in an aqueous solution. They will test it for mechanical disruption, where you see how long it takes for a kernel to be, or a sample of kernels to be ground up. They'll also look at kernel opacity and its effect on rheology, which is kind of a movement of mass. It's a little complex for this seminar, so I don't want to get too off in the weeds on it. But it's sort of think of it as a measurement of thickness and how hard it is to, say, stir a solution of it. Now, the problem is that none of these traits directly measure masa quality, such as moisture uptake and pericarp retention. They're all proxies. And looking around online, I saw that there really weren't many standards that exist for measuring corn properties related to food grade specific traits. So there are instances such as determining the amount of amylose or amylopectin in corn, or the amount of gluten, but nothing related to cooking qualities. And this is found from the Cereal and Grains Association and the Food and Ag Organization. So since there are no standards for measuring moisture uptake and pericarp retention, one of the primary objectives of my research is to develop standards to measure moisture uptake and pericarp retention in a high throughput manner. And when we do this, we'll hopefully be able to propose methods to either the Cereal and Grains Association or the Food and Ag Organization to have a standard test that breeders can use. And this will be extremely beneficial to breeders because it will benefit, or it will help them in multi-trait selection breeding, where they're going to want to maximize yield while also keeping the moisture uptake or pericarp retention within a specific range of values. And so having a quantitative measure will greatly benefit them in being able to release high quality lines quicker, or even create or even release a wider variety of these lines. And this will obviously benefit the growers, as they'll have more options to grow, as well as the fact that they won't have to worry about things such as dockages when they take their samples in to a food grade corn specific elevator. This lastly comes around to the manufacturer, as they will be able to have more confidence in their corn as they cook it, potentially having to adjust parameters less, because they can be more certain that their corn is in this just right range. Now, as I said before, one of the objectives of my research is to develop standards to measure moisture uptake and pericarp retention in a high throughput manner. So in order to do this, we need a diverse set of lines for analysis. We are choosing to use the Wisconsin Diversity Panel, as it's a phenotypically diverse set of lines. And the panel started in around 2011, so it's relatively new, but the big thing about it is it's a bunch of lines that, as I said, are phenotypically diverse, but still will grow in the Midwest. And so here at the U, we actually grow a subset of this, about approximately 500 lines. And using WiDiv is really beneficial to us because it actually has a lot of genetic information already set and ready to go. So it's really primed for genomics work. So first, in order to be able to answer many biological questions, we need to have a high throughput method to screen large numbers of genotypes for moisture uptake during nixtamalization. So I want to go into a little bit about how what things affect moisture uptake, and then later after that we'll talk about the pipeline, and then get into pericarp retention. So moisture uptake is a very complex trait, and it depends a lot on the composition of the kernels. If you notice the kernel in the upper right of the screen, I will actually use that to indicate what part of the kernel we're referencing. And so in this case, for the first instance, we're actually talking about cooking parameters, so it will affect the entire kernel. The various cooking parameters that can affect it are things like the cooking time or how long it cooks. It can also be affected by the steeping temperature or the amount of time it's left to steep. It can also be affected by pH. These are the primary components that people talk about in literature. Oftentimes, they are more concerned about how the kernel or how the cooking parameters are set up in a way to affect moisture uptake rather than composition. But this really doesn't put into perspective the composition of kernels, where things like the starch structure can greatly affect moisture uptake. For example, on top we see an amylose strand, which is sort of a linear starch molecule. Down below is amylopectin, which is more branched. And as you can see, it forms these little helices, both here on the amylose and here in the amylopectin. And these helices are really hydrophilic. Water really enjoys getting into the middle of those helices. And as you can see, amylopectin has a lot more helices and thus a lot more space for water to be absorbed into. And so the ratio of amylose and amylopectin could have a large effect on moisture uptake. You also have things like the pericarp, and so the composition of that and the thickness of it play a big role. I don't want to get too into the weeds on what all these different colors represent. It's mostly to show the large abundance of different macromolecules that are present in the pericarp. There's also interactions between different molecules. And so this picture here is a scanning electron micrograph of the starch protein matrix. And so the large granules that you see are are starches, and the small granules are proteins. And so this interaction is really important because, as you might be able to imagine, as the starch begins to absorb water and start swelling, the protein matrix is actually going to restrict how much it can swell. And so in theory, protein might have a slightly negative effect on moisture uptake. Now, in order, as I said in that last slide, composition is very important to moisture uptake. And we need a tool that will allow us to determine the composition of our different traits. And near-infrared spectroscopy is an extremely valuable tool in doing this. Just a brief overview, near-infrared spectroscopy is a method of scanning the electromagnetic spectrum specifically for the near-infrared region, approximately 700 to 2,500 nanometers. And we collect the absorbance values at these different wave bands. And so an important distinction here is we will actually be using wave bands. The machine we use collects 141 wave bands. You may be used to hearing the term wavelength, referring to, say, a wavelength of 1100. In our case, it collects every fifth waveband and sort of averages the nearby wavebands absorbances into the one it returns. So if it returns 1100, it might also be picking up a little bit of 998 as well as 1102. So companies such as Perton will actually have equations set up for determining composition where they take a sample of corn and they split it in two and they scan one part of it with an NIR and send the other part through some wet lab assays. Now, when you combine the waveband absorbances with the wet lab assay results, you can actually put them together to create a model. And Perton has proprietary models that they use to predict their different compositions. And these are very robust models. They train them on thousands of samples, maybe even tens of thousands of samples, and they create these strong correlations between the prediction and the actual trait value. And so we have high confidence in these values. Now, in theory, we should be able to do this with moisture uptake on our with our own samples. And so we have scanned many samples and done a wet lab assay, which I'll describe coming up. And putting these two together, we have created our own model, not necessarily the same as what Perton uses, but we have created our own model to predict these values. Now, in order to do this, I need to describe how we collected these lines for training. As I mentioned before, we have these 500 lines from the Wisconsin Diversity Panel that we grow at the U of M. We collected samples from each plot and scanned them with an NIR. We then used Honig's regression to determine the 100 most spectrally diverse lines present. We then the following year grew, or sorry, not the following year, but from those 500 lines, we grew them in 3 different locations across 2 years. Minnesota and Iowa were grown in 2016 and 2017, whereas Missouri was only grown in 2017. Now, these 100 spectrally diverse lines were grown among the full 500 at all 3 of those locations. But we really focus on the 100 spectrally diverse for training our model. This is our training set. This is what we'll be using to collect waveband absorbances and wet lab assays from. So we took a plot and we split it into 2 samples. And as I go along here, I will be showing industrial samples to show you how our scaled down benchtop cook test relates back to the industrial scale. So we split the samples and we combine it with a lime solution inside a cooking basket. And we heat these samples up on a hot plate. And we do this up to above the starch gelatinization temperature, but below water boiling point. and then allow it to steep in a hot water bath overnight, approximately 16 hours. These samples then have the pericarp removed. We do not remove it with a water jet. We remove it by hand rubbing the kernels to remove any excess pericarp. And then finally, we dry the samples, and the loss in mass from the kernels when they are while they're in the dryer is the loss of moisture, and so we can then determine the percent moisture they were at following the cook. Following this, the other sample from the plot is scanned with an NIR, and both the waveband absorbances and the moisture uptake are fed into a machine learning model. Now, before we go too much further, we really need to talk about some of the theory behind machine learning. So I want to run you through just some of the basics of machine learning so that you understand the terms that I'll be using later on in this presentation. We start out with a master data set, and we split this into a training and testing set. This is called a partitioning rate, and so depending on the data set or the type of data you're using, you can have a different value. An 80-20 split is very common, where you send 80% of your data into the training set and 20 into the testing set. It's very important that this testing set stays sort of enclosed in its own bubble and doesn't get to see what's happening with the training, so that we can make sure that the model isn't skewed in any way, shape, or form. Now, the training data set is often pre-processed. These are processes such as normalization or centering and scaling the data set. And finally, we use this pre-processed data set to train a model. Now, once we have a model trained, we can finally bring in the testing data set and test the model and test its performance. It's important that we pre-process this data set, this testing data set, the same way we pre-process the training set, so as not to clue in the model into what is within the testing set or make it too terribly different. Sorry, I lost my train of thought. It's important that this testing data set comes from the original data set, because it has actual, it should have actual values. It should have an actual value for the trait that we're trying to predict. And so then we can determine its performance either through a correlation or through a measure of error. And so going a little bit more in depth into machine learning, there's a process called cross-validation, where we take our training set and split it into different folds. In this case, we have 5 folds. And as you can see, it's split into these 5 folds. 1 is used for validation, in this case, the blue, and 4 are used, the rest, in this case, 4 are used as a training set. In this case, the training are the white blocks. And cross-validation will continue through until each fold is successively used as the validation set, and it can then take an average of these performance values to determine the performance of the model. And so if the cross-validation shows a good performance and the test model shows a good performance, we're sitting really well. The test or the model is performing well on data it's never seen before. However, if testing the model turns out to give a bad performance while the cross-validation gave a good performance, this likely means that we've overfit the model, and we either need to consider using fewer features or a more complex model. Another important piece of information to note is that we can adjust the hyperparameters. And so a hyperparameter, I'll actually use an example of an SVM, a support vector machine, to describe this. If we have a data set such as this, where we want to be able to split the blue stars from the red circles, we need to determine how important it is that we really describe or really separate them fully. So SVMs have a hyperparameter called C, which stands for cost. And so we're wondering how important, what the cost is of misrepresenting that blue star that's over by the circles. And so if it's really important we predict everything right with the blue stars, we might get a line such as this to split the data set. Whereas if we say it's really important that we don't accidentally predict a red circle as a blue star, we might have a plane such as this, where we say we can miss one or two blue stars. And so these hyperparameters are really important for training a model, as it will affect how the model interacts with the data before it's even seen the data. So fortunately enough for us, R has a very robust package called Caret, which has over 100 models to choose from, and allows for a high degree of customization of the models, which is very beneficial, as one model or one setting within a model is not necessarily going to be good for all sorts of different data sets. So coming back from the theoretical framework of how machine learning works, I want to talk to you about how we're actually implementing it, how we're using it with the spectral data. So spectral data provides a lot of information. As you can see here, there's a wide range of possible values that these different wavebands can take. They're often highly correlated, although different. As you can see, there are differences on the y-axis. And this really led us to suspect that we should normalize the spectra, as it would not be good to see two samples that are similar to each other but have moderately different spectral values. We don't want to see two moderately different spectral values having close actual moisture uptake values and then being predicted very, very differently. And so when we normalized these, we had 4 different normalizations that we used. This plot in the upper left shows the base spectra that was just on the screen. We still use this as sort of a baseline to understand how the models performed. But as you can see, there's a wide range of emphasis on different features. If we look at the peaks here around just shy of 1500 nanometers, we can see that there's quite a range of importance of them. And by importance, we're looking at things of how much it spreads out. So if there's not much spread to it, it might not be as important, and so the model won't be able to differentiate samples very well off of it. We also get areas such as this, just shy of 1300 nanometers, where there's not as much spread of value, but there is still some range in value. And so in order to look at these and build models, we have to come up with a list of models we want to use. In machine learning, there's a common phrase saying there is no free lunch. And this means that a model that works well on one data set or in one situation might not work the best on another data set. And so it's usually a good idea to do some literature review and see what kind of models are commonly used on your type of data. Ideally, in an ideal world, we would test all the models we can get our hands on, but realistically, it's just not that feasible with computational power and things like that. So we focused on these specific models. We looked at principal component regression and partial least squares regression. We also use support vector machines and a variety of kernels. A kernel is a variation of a support vector machine. We also used random forests, and these were, of course, all in the Caret package. So we have 7 different models and a number of other variables to test. The hyperparameter, we had 141 hyperparameters, 1 for each waveband, if you remember me mentioning that earlier. We can partitioned by two different features. We can either, when we had originally split the data set into training and testing, we can either split it randomly or by a group such as the genotype, which is what we did. You can also cross-validate by either randomly or by a group such as genotype. And we also had the 5 Spectra normalizations that we just covered. This leads to just under 20,000 total combinations of different settings to try. And we wanted to bootstrap this, meaning we wanted to repeatedly run these models 100 times. And this was to make sure that just because one model came out as the best, we wanted to make sure that that wasn't just a random happenstance and we didn't get fooled by the computer. And so bootstrapping this 100 times gave us just under 20 million iterations to run through, which is a lot to put on a standard laptop computer. And so we actually utilized some parallel computation using the Minnesota Supercomputing Institute. And so our output ended up looking similar to this. This is just the top 5 models. As you can see, they all perform very, very similarly. You actually have to go out to the 4th decimal place just to see a difference. And I believe you actually have to go out to about the 20th model combination to see it drop below 0.7. And so we decided to still, went the wrong way. We decided to still stick with this first model, even though they're all so similar. We decided to stick with this first model because it would give us the largest chance at producing a strong model that we can still obtain a lot of information from. And so if you remember this plot from before, we had those 500 lines grown at these 3 locations across 2 years. This gave us 5,000 different plots that we could validate our model on. All of these plots were scanned with an NIR, and with that, we then fed it into this machine learning model after the 100-line training set. So this model gave us moisture uptake predictions from these 5,000 data points, and we plotted the distribution of these predictions to make sure it seemed somewhat normal and reasonable for that matter. One thing to note here is that the black filled in bars on this histogram are samples that fell within the training set. And so our training set went from about 0.37 for moisture uptake value to about 0.52. Anything outside of that is the empty white bars, just simply meaning that they were extrapolated by the model. We didn't have a sample beyond that to really compare the waveband absorbances to. And so we decided to take 48 samples throughout this distribution and cook them in our scaled down benchtop cook test to really see how well our model was actually predicting. And when we look at the performance, if we look at the correlation between the predicted moisture uptake and the actual moisture uptake, it looks good if we remember to not trust the extrapolated values. With linear regression, we typically don't want to, with regression, we want to be careful with extrapolated values because we don't have anything to really compare it to. One extreme value could really throw off the entire equation. And so we decided to base this off of only the values that fell within the training set bounds. And so the rationale or the logic behind doing this is that we can then, when we release this model, we can actually add a line of code that says, if it's below or above these bounds, just say it's not trustworthy. And that is, we feel that's more than acceptable because the values that they're going to be looking for are right around that 0.45 range. And as we can see, the model predicts very well within that range. And so we actually had a Spearman R value of 0.85. And so that's going to be looking at the relative rank of these individuals. And the purpose of doing that is, compared to going with a, say, a Pearson R correlation, is that if the errors are relatively high, we can still be confident in saying that we want, or if we cook a value at the upper limit and a value at the lower limit of what's acceptable for Masa quality, we can, as long as the model predicts within this range, we can be confident in telling the breeders that yes, this will be a strong value, or this will be a strong contender. It should very well perform as expected. And just a note here, the dotted line in the background of this graph is just showing a 1-to-1 ratio. And so we want this black line to be as close to the dotted line as possible. And this is actually performing very well. If we added in the empty dots, it would be more of a horizontal line, which is getting away from our 1-to-1 ratio. So what does this mean for an industrial pipeline? It means that the manufacturers can take samples, scan them, feed them into the machine learning model, and get moisture uptake predictions without ever cooking a kernel. So they don't have to go through the roughly 18-hour, well, probably more about 18 to 20-hour cooking process just to determine what the moisture uptake will be. So I just want to switch gears a little bit and briefly talk about my Pericarp retention project, where the purpose is to develop a high throughput method to, again, screen large numbers of genotypes for Pericarp retention after nixtamalization for these large scale breeding programs. And just to go back to kind of what Pericarp is, to remind you, it's that layer of tissue surrounding the kernel. It's made up of a number of different substances, primarily fiber and protein, some phenolic cross-linkers, things like that. And it's a very complex lining around this kernel. Now, the pericarp should largely be removed during cooking. And if you can see it in the picture off to the right, there's sort of a line right here where you can see the pericarp sitting in the bottom of this beaker. The issue is that if you take a closer look at some of these kernels, you can actually see not all the pericarp is removed. In these cases, this amount of pericarp would likely be okay, but there are often times where kernels will retain pericarp along the entire edge of the kernel and not just hanging off the tip cap. And so that is where more of the problem can come in. And so in 1991, Cerna Saldivar released a paper describing a process to actually evaluate the amount of pericarp retained. And so this is a process where they go through and they boil the samples in a lime solution. They quench it to stop the cooking process, and then they'll stain it in this May-Gruenwald solution and clean it in methanol. They then take these stained kernels and they visually evaluate them with a subjective score of 1 to 5. And I really want to emphasize that this isn't the best method to be using in terms of breeding. It doesn't give a very large dynamic range of values, and as it says, it's a subjective score. One person to the next might score kernels differently. And so we want to try to remove that source of error from this process and make it more high throughput. And so what we're proposing to do is take this whole method up until the stained kernels and take pictures of these kernels after they've been stained. And as you can see, the stain anywhere with a purple or green or bluish hue is areas where the pericarp is left. The differences in color is theorized to come from sort of the thickness of the pericarp, though that is something that will need to be looked into. And so we should be able to write a computer program to do edge detection for these differences in color and say, find an edge where the color looks blue or purple or just not the same color as the kernel. We can fill in this area and determine the amount of kernel that is covered by Pericarp still and get an actual quantitative value of this. And so this is the pipeline that we'll be proposing for the Pericarp retention. And now that I've shown you both the moisture uptake pipeline and the Pericarp retention pipeline, I want to just briefly go over how we're going to utilize these pipelines to answer biological questions. So we'll use these to perform genetic dissection of the traits, where if you remember, I told you that the Wisconsin Diversity Panel is primed for genetic work with the large amount of genetic information readily available. And so we can use these to perform a genome-wide association survey, where we'll take different SNPs, single nucleotide polymorphisms, and correlate them to the different trait values. This plot here is a Manhattan plot. It's a very common plot to make for these genome-wide association surveys. And really each data point is a SNP, and the higher the data point is, especially if it's above that horizontal line, it signifies the strength of the correlation between the SNP and the trait. And so anything above that line is considered a significant correlation. We also want to assess the utility in a hybrid system. This is primarily for the moisture uptake, as the Pericarp retention is a computer vision model, which should be just fine since it's essentially just taking pictures and looking for color. And so the reason for doing this is because hybrids and inbreds perform very differently. And so as you can see, the inbred parents on the side seem to have a much lower performance than the hybrid in the middle. And this can be seen in the kernels or the cobs as well. And so we want to be able to make sure that even with these differences in performance, that the model will still work in a hybrid system, since breeders are releasing hybrid corn, not inbreds. To do this, we have multiple genotypes from breeding companies that were at late stage testing and grown in multi-environment trials. We're going to scan these samples and put them through the cooking process again, and compare the predictions between from the machine learning model and the cook test values. In theory, a strong correlation should mean that the model is transferable to a hybrid system. Lastly, we want to determine sources of variation. And so we'll be again using the same late stage testing genotypes in a multi-environment trial, the same ones as the last objective, to look at how the genotype, the environment, and the genotype by environment interaction affect this. And so the reason for doing this is because different genotypes grown in different environments can perform very differently. And so as you can see here, genotype 1, in environment 1, the first genotype, or the two genotypes, perform very differently, and in environment 2, they actually switched which one was the top performing genotype. And so this is actually a very common and well-known phenomenon. It's the equation used to describe this is that phenotype equals genotype plus environment plus the genotype by environment interaction plus some source of error. And the issue with just looking at only the environment and saying, well, this one was grown in Iowa, this one was grown in Minnesota, is you don't account for things like directly account for things like soil or the weather or the amount of competition, kind of how densely the plants are planted next to each other. And so this can largely affect the performance of the models. And we want to know how the genotype, environment, and G by E affect the moisture uptake and the Pericarp retention traits. And so if we know how much environment plays a role, we can actually start looking at what environments are optimized for food grade corn. And so early results show that environment does play a really large role, at least in inbreds. And so these were grown in the 5 locations described earlier. And as you can see, environment plays a very large role. Just to quickly summarize what we've talked about, because it has been a lot. Corn is very important, and food grade corn, while it makes up a small percentage, is still a large acreage and worth a lot of money to the industry. And we want to develop methods to efficiently screen samples in a high throughput way for breeding companies to be able to better utilize. And we want to address multiple biological questions as to the effects underlying these both of these traits. I just wanted to take a moment to thank my advisor, Candy Hirsch, everyone in the Hirsch lab, Rafa, Jonathan, Dorothy, Claire, Yinji, and Amanda. And I also wanted to thank Mark Holmes, who did a lot of the sample collection and cooking, and George Anor in food science for his help with a lot of the food chemistry, and Nick, Dave, and Mandy from PepsiCo for a lot of their intellectual input on these projects. And last but of course not least, I want to thank PepsiCo for funding this presentation. With that, I'll take any questions there might be.
So if anyone has any questions, there are two ways you can go about asking those. The first is there's a Q&A button at the bottom of the screen, and if you click that, it'll bring up a window where I think you can type in a question, and then I can read it and Michael can answer it. Or if you'd like to ask him your question directly, there is a raise hand button, and Cody can unlock you so that you can ask your question directly. So Michael, as we're waiting for the first questions to come in, I have a question for you about the cooking process you were talking about. And you mentioned that in your scenario, you are removing that pericarp by hand, and in an industry scenario, they were pressure washing it, I think you said.
Yep.
How do you make sure that in removing it by hand, you're getting, about equivalent as they would in that industry?
Yeah, so great question. We actually, it's a very long pericarp removal process. We will wash the kernels with water. We'll rub them between our hands approximately like 20-ish times. We'll then decant off any of the water, add the kernels back to the bowl, add water, rub them again, and we'll do this about 3 or 4 times. And we've worked very closely with some people at PepsiCo to make sure that this benchtop cook test performs approximately the same as the large scale cook tests.
Okay.
It looks like Dorothy has a question. Go ahead, Dorothy.
Hello?
Yes, we can hear you.
Okay, great. I have a question about slide 75. You were talking about the 100 spectrally diverse lines that you chose for your training set. Could you elaborate a little bit more about what you mean by spectrally diverse and how those were chosen?
Yeah, so we had, as I said, we had those 500 lines. We scanned them and we used a process called Honig's regression, which from my understanding is a process of saying what is the maximum of absorbance that we find among all the samples. Okay, it's sample 18, we'll select that. We'll then subtract that from all of the samples, that absorbance value from all of the samples. And then we'll look at what is the most extreme value again, take that out, and again, subtract its absorbance values. And so Honig's regression is just a method of determining, finding a highly representative sample. In this case, what would be spectrally diverse samples for us.
Great, thank you.
Mm-hmm.
Okay, so Michael, Jerry Cohen has a question, and he asked if, since Okay, sorry, let me start again. He says, your slides on NIR are labeled absorbance, but his understanding is that typically those systems measure diffuse reflection. If so, how would changes in the physical properties of the powder change in contrast to changes in the chemical composition?
I'm not sure. I'm just gonna read the real quick.
It was a long one.
Yeah, so the from my understanding, the machines do measure in reflectance, and there's an equation to then convert it to absorbance. And so the changes in the physical properties of the powder Jerry, if you're referring to the physical properties such as the size of the powder, we grind all of our samples to a uniform size and scan them. But so the big changes should primarily be the chemical composition, which will then change based on the absorbance, the amount of that waveband absorbed by the kernels. Jerry, if I didn't get your question, if I didn't answer exactly what you were looking for, send me an email and I can try to respond to it.
Okay, great. And Ethan also has a question, and he asks, will the development of the Masa quality prediction model allow for the industry to blend batches of corn, meaning combine the high and low moisture retaining batches?
Yeah, so that's something that in theory, the answer should be yes. That's going to be something else we're going to have to look into. The next big step is determining if it works with hybrids. But yeah, so in theory, all of this should be based largely off of the chemical composition of the kernels. And so assuming that the batches that they use in industry and the mixed batches aren't outrageously different from the samples they were trained on chemically, they should perform similarly. But yes, that is something that we'll have to consider after we determine that it works on a hybrid system.
There's also a question from Tess Anderson. I will allow her to, or them to ask it now. Tess can.
So I was wondering, with all the work that you've done with these projects, what is next?
More projects. To be totally honest, I haven't been looking terribly far ahead. I hope to continue on and finish my PhD. Beyond that, I'm hoping to get a job in industry. But that will, of course, all be dependent on how things like student teaching and things like that go.
Does anyone have any more questions for Michael? If we can just wait another minute to make sure that everyone's questions are answered before wrapping things up. So it looks like Jerry has another technical question.
I'm reading it now.
Okay, I can read it aloud, just I don't think everyone else can see it. So Jerry asked, would it not be better to use oh man, these are words I'm not used to saying, monochromatic LED illumination for your stained kernels rather than using the less precise color from a typical image to define the stained regions?
Quite possibly. A lot of the Pericarp retention project is still yet to come. I am in very early stages of planning it out. We do not have a computer or an image capture system set up yet, nor a computer vision model or anything. That this was all, everything related to Pericarp retention was more a proposal. But I will email you and talk to you about this, because it would be nice if we didn't have to consider the differential colors that the staining process would create.
that is the last question from the audience. I think we can thank Michael and wrap things up. Thanks for a great presentation.